Welcome to our lecture on genetic testing and diagnosis, focusing on the intricate processes and advancements in this field. Pre-implantation genetic diagnosis, or PGD, represents the earliest form of prenatal testing, offering insights before implantation. PGD is a technique used during IVF to test embryos for genetic disorders before transfer, allowing for informed decisions. PGD matters because it allows for early selection, reducing difficult decisions later and helping carriers of genetic conditions. The PGD process involves several steps, from ovarian stimulation to embryo transfer, ensuring thorough genetic analysis. This diagram illustrates the PGD process, from egg retrieval to embryo transfer, highlighting each critical step. New terminology in genetic testing helps clarify what is tested and who benefits, enhancing understanding and communication. The history of PGTA shows its evolution and the advancements in genetic testing over the years. Technologies like NGS, PCR, and FISH are crucial in genetic testing, each offering unique advantages. Limitations and ethical considerations in PGD include mosaicism, allele dropout, and ethical controversies. Genetic testing spans from embryo to adulthood, covering various stages and purposes. By the end of this lecture, you'll be able to define, explain, apply, and recognize key aspects of genetic testing. Accurate genetic diagnosis provides precise answers, guides management, informs decisions, and enables cascade testing. Genetic testing categories include diagnostic, newborn screening, carrier, and predictive testing, each serving unique purposes. Newborn screening detects diseases early, using blood samples to identify potential metabolic errors. Explore the diseases screened in newborns through the UAE DOH brochure, highlighting key conditions. Diagnostic genetic testing involves a structured approach, from clinical evaluation to genetic counseling. Different technologies in genetic testing offer varying resolutions, from karyotype to whole exome sequencing. Karyotyping visualizes chromosomes, detecting numerical and structural abnormalities with a specific resolution. Fluorescence in situ hybridization, or FISH, uses fluorescently labeled DNA probes to target specific chromosomal sequences. This technique is crucial for identifying deletions, duplications, and other chromosomal abnormalities. It provides rapid results, typically within 24 to 48 hours, making it invaluable for prenatal testing and cancer diagnostics. However, FISH is limited to detecting only what it is designed for, which means it might miss some mutations. Chromosomal microarray analysis, or CMA, is a powerful tool for detecting copy number variants across the genome. It offers a resolution much higher than traditional karyotyping, making it ideal for identifying submicroscopic deletions and duplications. CMA is particularly useful for diagnosing unexplained developmental delays and congenital anomalies. It can also provide answers when karyotyping fails due to cell culture issues. Sequencing-based tests such as targeted gene panels, whole exome sequencing, and whole genome sequencing offer varying scopes and diagnostic yields. These tests are crucial for identifying point mutations, small insertions, deletions, and splice site variants. Each test has its advantages and limitations, making them suitable for different clinical scenarios. Choosing the right genetic test depends on the clinical question and the type of abnormality being investigated. Whether it's a chromosomal abnormality, a specific gene mutation, or a broader genomic issue, selecting the appropriate test is crucial for accurate diagnosis and treatment planning. Carrier testing is a genetic screening for individuals planning a family. It helps determine if they carry genes for recessive or X-linked disorders, such as cystic fibrosis or sickle cell disease. This information is vital for making informed reproductive choices, especially if both partners are carriers, as there is a 25% risk of having an affected child. Predictive genetic testing is used to identify individuals at risk for conditions before symptoms appear. This testing can guide management decisions, such as increased surveillance or preventive measures. However, it also raises ethical considerations, especially when no actionable treatment is available. Ethical considerations in genetic testing are paramount. Informed consent, the right not to know, and the handling of incidental findings are critical aspects. Additionally, issues like genetic discrimination and testing of minors require careful navigation to protect individuals' rights and privacy. Thank you for your attention and interest in genetic testing. We hope this presentation has provided valuable insights into the various testing methods and their implications.